Welcome to the deep dive. Our mission today is really laser focused. We are looking past the general hype, the abstract promises of AI, and we're diving right into the scientific trenches where it is already operating as a true co-scientist. We're looking at where these systems are tackling some of the highest stakes, most complex challenges on the planet.
And the stakes really couldn't be higher, especially when you talk about drug discovery.
Oh, absolutely.
For anyone in pharma or biotech, you just know the brutal inefficiency of it all. Up to 90% of all drug candidates, they fail. They fail in clinical trials.
90%. That number is just staggering. And that failure, it so often traces back to the very first step, right?
It does.
If you get that foundational work wrong, identifying the right biological target,
then everything downstream is compromised. The compound, the trials, the billions of dollars, it's all built on a shaky foundation.
Exactly.
So our deep dive today is structured around how AI is starting to fix this catastrophic failure rate. We've got two key tracks.
Okay.
First, we're going to look at some dramatic efficiency gains through specialized models. We've got a fantastic story about liver disease as our backbone for that.
And the second track.
Second, we'll examine how science is building, rigorous frameworks to actually ensure we can trust these complex AI predictions.
Right, because you need actionable intelligence you can stake your research on,
Yeah.
not just impressive stats.
Precisely. From predicting molecular structures all the way to accelerating fundamental research by, believe it or not, a factor of 1,000.
1,000. Okay, let's start with that success story then, the one that sort of defines this future blueprint. You mentioned liver fibrosis.
Yeah, liver fibrosis. It's a massive silent killer. We're talking about 1 million deaths globally every year.
Wow.
It's essentially scarring of the liver. It's driven by these activated cells called myofibroblasts that just deposit huge damaging amounts of collagen.
And the research on this has been stuck for a while, I understand. The testing ground itself was flawed.
Exactly. We were relying on animal models that, they often don't replicate human fibrosis accurately.
So a dead end from the start.
Or we used these older flat dish models that just didn't have the right mix of human liver cells. They couldn't reproduce those thick, damaging collagen filaments you see in actual human tissue.
So if your model's wrong, your drug candidates will be wrong.
You get false positives that fail instantly in humans. It's a huge waste.
Okay, so the first critical step here wasn't even the AI. It was fixing the experiment. They built a better platform.
They did. They developed these multi-lineage human hepatic organoids, or microHOs, grown in microwells. And these are special because they contain the correct mix of human liver cells. And crucially, when you expose them to damage signals, they actually reproduce the disease. They form the myofibroblasts, the dense scarring, everything.
So you have a high-fidelity platform, a reliable test bed.
The test bed that drug screening desperately needed.
Yes. And then they brought in the AI co-scientist.
Then they unleashed the AI. And this wasn't some general LLM just, guessing answers. This was a highly specialized, multi-agent AI system.
It built on Gemini 2.0, I saw.
That's right. And it was tasked specifically with hypothesis generation.
So not just finding patterns, but proposing new ideas.
Proposing new ideas within a very targeted mission, they told it. Find antifibrotic drugs, but only ones that work by tweaking what are called epigenomic modifiers.
Okay, break that down for us. Epigenomic modifiers.
Think of them like master dimmer switches on your DNA. Things like HDACs and BRD4, they control how often and how strongly a specific gene gets expressed.
So the AI was looking for which dimmer switch to turn off to stop the scarring.
Exactly. And the results were striking. Out of 25 drugs they tested on these micro-Os, 5 showed significant antifibrotic effects.
Okay, 5 winners.
But here's the kicker. 2 of those 5 winners were exactly the types the AI had recommended, an HDAC inhibitor and a BRD4 inhibitor.
And what about the ones the human scientists picked?
And this is where the human bias check comes in. The targets picked by human scientists, based on all the existing well-published literature, things like EZH2 and SMARCA24 inhibitors, they failed. Completely failed in this high-fidelity platform.
Wait, so the established research path, the consensus built on those older, flawed models, it led experts down the wrong road.
It absolutely suggests that. The AI cut through the noise. It ignored a human consensus that might have just been confirmation bias from bad data.
It found a signal everyone else had missed.
Exactly. It leveraged data pointing to a mechanism that humans had either overlooked or just deemed less important. That is the power here.
And the practical outcome is huge. The lead candidate, The one the AI recommended.
An HDAC inhibitor called vinostat, or SAHA. And get this, it's already FDA approved as an anti-cancer drug.
So it's a repurposed drug. That's a massive advantage.
Phenomenal. It dramatically de-risks and accelerates the whole timeline, and the mechanism is fascinating. It didn't just slow the disease, it rewired the cell's entire fate.
The 91% reduction in chromatin changes, right?
91%. It wasn't a simple brake on a pathway, it was a steering wheel.
A steering wheel, not a brake. I like that.
It literally prevents the key driver cells, the hepatic stellate cells, from ever transforming into those problematic myofibroblasts in the first place. It steers the cell away from becoming a problem.
That is the blueprint. Specialized AI, rigorous model. But it immediately brings up a huge question.
I know where you're going with this.
If every lab, every pharma company builds its own proprietary specialized AI, how on earth do we compare them? How do we build trust across the entire industry?
That is the problem, the benchmarking gap. You can't trust a black box. You need an objective evaluation system.
Which is where this other work comes in, Target Pro and Target Bench 1.0.
Exactly. This is the integrated framework designed to solve that trust problem. Target Pro is the machine learning workflow, and Target Bench is the objective evaluation system.
And Target Pro isn't just predicting if a molecule might bind to something, is it? It's trying to predict something much, much more valuable.
It's predicting clinical success. Will a target move forward from phase I trials and beyond? That is the highest stakes prediction you can possibly make.
And to do that, they're feeding it all kinds of data. It's multimodal.
Very. 22 different input scores, everything from deep biological data omics, gene expression, to textual features like scientific literature attention scores, even grant funding levels.
And what's really illuminating is how the model decides what to listen to, the feature importance, right? It changes.
It's context aware. It depends entirely on the disease. For cancer and fibrotic diseases, the raw biology, the omics data was the most important thing, about 65% of the predictive power.
Because those are often driven by very specific genetic mechanisms.
The raw data is pinged there. But then you look at the more complex systemic conditions, immune, metabolic, neurological diseases.
The consensus flips.
Completely. For those, the text-specific features become dominant, more than 50% importance. It's like the AI knows that for diseases we've studied for decades, the accumulated human consensus in the literature is actually a stronger predictor of clinical success than finding one brand new genetic pathway.
That's incredibly sophisticated. So the big question, how did it do? How did Target Pro perform against the big generalist models we all hear about?
This is the headline result.
Yeah.
This is what shows the power of specialization. Target Pro achieved an overall clinical target retrieval rate of 71.6%.
71.6%. And how does that compare to the generalist LLMs?
It's a 2 to 3-fold improvement. The generalist models, they only managed rates between, say, 15 and 40%.
So a huge gap.
A huge gap. The specialized AI, trained on what actually works in the clinic, not just what's biologically plausible, it's winning.
And the targets it finds are actually actionable. They're ready to go.
Precisely. For example, nearly 96% of the novel targets it predicted already had a protein crystal structure available. That's a massive shortcut.
And repurposing potential.
Huge. 46% already had approved drugs for other diseases, just like the vinostat example. Very high repurposing potential.
Okay, so that confidence in target selection is paramount. But you also need confidence in the physical representation of the target itself. Which brings us to structural biology.
And the hallucination paradox.
Yes, explain that. Models like AlphaFold 3 can create these structures that look good, have a high confidence score, but are actually wrong.
It's a huge danger. It's a high confidence, low accuracy structural hallucination. The model gives you a beautiful looking protein complex, says it's 99% confident, but the structure is physically impossible. Atoms are clashing. It's junk.
And if you start designing a drug based on that fake structure,
you've wasted months, maybe years, and millions of dollars.
So we need a way to check the model's work from the inside. This is where code comes in, chain of diffusion embeddings.
Right. It quantifies something they call topological frustration.
That's a great phrase.
Isn't it? It basically means the energetically conflicted regions, the structural constraints, deep inside the model's own processing. Code looks for where the different parts of the structure are fighting each other.
So PLDDT, the old score, tells you about overall stability. But code tells you where the stress fractures are.
That's a perfect way to put it, and it's much better at it. Code had a correlation of 0.36 with the number of atomic clashes. That's a 100% improvement over PLDDT, which was only at 0.18.
So you use them together. One tells you if it looks right, the other tells you if it makes sense.
And it's incredibly practical. Using this framework called CONFIDE, they can identify the critical working parts of enzymes.
Zero shot, I read. No extra training needed.
No training. They just rank the parts of the enzyme with the lowest code scores because a functional site has to be rigid and stable. That simple ranking found the right spot 83% of the time. It's a scalable way to figure out what millions of unknown enzymes actually do.
Okay, so we've covered the specialized, benchmarked AI. Let's pivot now to the other side of the coin. The frontier generalist models, like a GPT-5, and how they act as, well, co-investigators.
Right, as these powerful, time-compressing partners in really fundamental theoretical research.
They accelerate the whole workflow.
The cosmic physics case study is a perfect example. A researcher was trying to derive these highly advanced mathematical structures, Lie point symmetries, for the wave equation around a Kerr black hole.
Sounds like something that would take a PhD thesis to solve.
It's very, very advanced stuff. And initially, the model failed, the classic cold start problem. It said no such symmetries existed.
But the researcher didn't just take that as an answer.
No, they used strategic scaffolding. They warmed up the model by giving it a simpler, flat space version of the problem first.
And that little bit of guidance was enough.
It was the key. After that warm-up, the model succeeded. It generated the correct, highly complex generators in about 18 minutes. It shows the human expert is still crucial for structuring the thinking process.
But the most dramatic acceleration example has to be the one from inertial confinement fusion, ICF modeling.
Oh, absolutely. Here, a researcher used GPT-5 to set up a reaction diffusion model for thermonuclear burn. Incredibly complex physics.
The kind of thing that takes a huge amount of time.
The researcher estimated the full workflow from concept to coding the model, running simulations, and then deriving the theory to explain it would normally take an expert about 6 person months.
6 months, and with the AI partner.
About 6 person hours.
That's a factor of 1,000 time compression. That's just wild.
It's unprecedented for this kind of work. And the LLM wasn't just, writing code. It verified its own results.
How so?
It derived a theoretical power balance criterion from the underlying physics that perfectly explained the optimal density profile its own numerical simulations had found. The theory validated the simulation.
That verification step is so important. The researcher noted you still need the expert eye to challenge the LLM, right? They can sometimes offer convenient solutions.
Numerical duct tape was the phrase he used. The human is still the critical thinker, the quality control. But even in pure abstract math, it's a catalyst.
Erda's problem, #848, a decades-old open problem.
And the LLM provided a key, high-level idea that led directly to the solution. A critical conceptual breakthrough. It's the ultimate co-investigator.
So if we pull all this together, what's the core lesson from this deep dive?
I think the future of science acceleration relies on an integrated system. You have to combine the highly specialized, rigorously benchmarked AI, your target pros, your confides,
The precision instruments?
with the frontier generalist models, your GPT-5s, serving as these powerful time-compressing co-investigators.
Speed and precision.
You need both to generate trustworthy, actionable intelligence.
That synthesis is really the key. But the ultimate vision here is it's even more ambitious than just those two parts working together.
It is. The provocative thought really is the idea of a fully automated, closed-loop discovery system, a self-driving lab.
So the specialized AI prioritizes the targets.
Robotic systems run the high-fidelity experiments, like the micros we talked about.
And the results instantly feed back to retrain and improve the AI models.
It's a continuous cycle. Prediction, testing, learning. It removes human bias and, crucially, the lag time completely. It's an intelligence loop.
The highest expression of the co-scientist model. A fascinating and maybe slightly scary potential endgame. And for you listening, if you are curious about these new standards for trust, that evaluation system, TargetBench 1.0, is actually public. It's open source.
That's right. You can go and check out your own research lists or see how your own pipeline compares against their metrics. The place to go is targetbench.org.
That's targetbench.org. Definitely worth exploring. Thank you for diving deep with us today.